All for joining us. This is an incredibly exciting day for David Julius, for the many people who have worked with him over the years, and for UCSF overall. In fact, we actually have two laureates today because David's colleague, Ardem Patapoutian, also was a postdoctoral scholar here at UCSF from 1996 to 2000 in the lab of Dr. Louis Reichart. Now, I've admired David's creativity, scientific excellence, and leadership over more than 3 decades at UCSF. He absolutely exemplifies the tenacious curiosity that drives the university's research mission and that can lead to exciting and unexpected advances for human health. As many of you know by now, David's work has been driven by the fundamental question, how do we feel pain and other sensations? His identification and characterization of the nerve cell ion channel receptors, known as the TRP receptors, led him down an intriguing scientific path in which he has employed natural products, including wasabi, scorpion venom, and more products such as that, in research that has fundamentally changed our understanding of pain and sensation. Remarkably, the TRPV1 receptor responds to either painful heat or capsaicin, the pepper that gives hot chilies their heat. Likewise, TRPM8 responds either to uncomfortably cold temperatures or to menthol. David and colleagues have identified a third TRP channel, TRPA1, that responds to the pungent chemical in mustards like wasabi, and it's also involved in inflammatory pain signals. In an absolutely scientific tour de force over the past several years, David and UCSF's Weifan Cheng have teamed up to use cryo-electron microscopy, a brand new technique to determine the structures of TRPV1 and TRPA1 at near atomic resolution, amongst other ongoing projects between their labs. All of these receptors play a critical role in normal sensory and pain processing, and they contribute to chronic pain syndromes caused by inflammation and tissue or nerve injury. Over the years, David has talked about how his fundamental physiologic research on this question was motivated early on by what he saw as a dearth of therapies to treat one of the most common and debilitating medical conditions, that is pain. Today, with pharmaceutical companies pursuing new pain therapies that target the TRP receptors, David's work has come full circle. Now it's my enormous pleasure to invite David to say a few words. Please accept our deepest congratulations, and let's begin by asking you, how does it feel to be woken up at 2 a.m. in the morning to learn that you've won a Nobel Prize?
Well, thank you so much, Sam, for that introduction. And first, I should say this is a great honor. And most importantly, it's really a great honor for all the trainees who worked with me and my colleagues. And I'm so proud to be here at UCSF and to have this recognition go to the University of California and particularly to our campus. Yeah, it's quite a thing to be woken up at 2 a.m. It's often difficult for the Nobel Committee to find you, and so they got me through some circuitous route. So I was sort of woken up not by the committee, but by a relative who said they were trying to get a hold of me. Anyhow, so that was interesting. And then, you know, things evolved from there. I knew within about 15 minutes, life was going to get pretty busy. So it's been a busy morning.
Robin, I'll turn it back to you to moderate the questions.
Okay, thank you, Sam. And the first question is from Issam Ahmed. And he would like to ask a live question. So unmute him. And just a reminder, if you can, to adjust your name on the screen. You can do that, I think, using your video function, so that we know the outlet it is with. Thanks.
Hi, can you hear me?
Yes. Good morning, Isa.
Good morning, Dr. Julius, and congratulations.
Thank you.
Thanks for doing this. I'm with AFP, Agence France Presse.
Mm-hmm.
I was wondering if you could please go back to the story of how you came to want to study capsicum. It came to you at a supermarket, I believe I read, and if you could tell us a little bit more about that, and how often do these sort of sort of scientific investigation arise from your sort of daily life observations? Is that why you pursued wasabi and menthol, for example?
Well, in part, I've when I decided I wanted to go into the area of neuroscience after my graduate work, I really got fascinated with the idea of how people interact with natural products in their environment. How do they learn to use plants for medicinal purposes, for ritualistic purposes, and how do they come to identify these plants, and then how do scientists use these plants in their products to really uncover basic biological mechanisms? So I was sort of already interested in that area. And then I got more and more interested in understanding pain mechanisms. And really the confluence of those two things is to look to plants that have chemical irritants that activate our pain pathway. That's presumably a protective mechanism for the plant. So those things came together. But yes, it's true that especially when you study sensory systems, which tells you all about the world that you live in, you're very aware of, you take lessons from life around you. And it is true that walking through the supermarket aisle one day, seeing all these hot chili pepper sauces, et cetera, I was thinking, we really have to get this project done. And my wife said, well, then you should get on it. As we were looking at these things, I was looking at all these spices. But you're right that, sensory systems, what we see and how we hear and how we sense temperature and pressure, these really tell us about how we interact with the world. So when we see things in the world around us, it begs these questions naturally, just out of curiosity.
So we have a similar question from Tina Say, who is with Science News Magazine. And Tina, I wanted to know if that answered your question, or do you have a follow-up question for that?
Well, yes, I was just kind of wondering too about what inspired you to use menthol, and did you know at the point when you were looking at these irritants that might cause pain that they would also be involved in heat and cold sensing?
Mm-hmm. Yeah, that's a great question. So when we went to look for the molecular mechanism of the receptor for capsaicin, we started that because we were building on earlier work from physiologists in Hungary who showed that capsaicin was an activator of neurons that are involved in pain sensation. What wasn't known was how this worked. But we did not know that would be the same mechanism that nerve fibers use to sense heat. And we only learned that after we identified the receptor for capsaicin. But after that, once we decided to look at menthol as a cooling agent, taking a lesson from capsaicin and heat and that relationship, we had a pretty good sense that if we found the receptor for menthol, that would tell us something about cold sensation. And again, that was building on work from physiologists in Sweden, Hensel and Zodeman, decades ago, who showed physiologically that there was some relationship between how menthol worked and how cold worked. So that particular aspect of it was not a surprise, but again, nobody really understood the molecular basis behind that.
Great. Do we have any other questions?
David, let me jump in with perhaps two questions. One of the amazing things about the kind of work that you and others do is that it's driven by innate curiosity, and at least at the beginning, not necessarily with a therapy in mind. Can you just expand a little bit on that the power of curiosity-driven research?
Yeah, I'd be happy to. I mean, that's something I talk about a lot. I think there are so many examples of this in medicine and in biology. Most of the great advances in at least in terms of understanding mechanism medicine really started off by people just following their curiosity and answering basic curiosity-driven questions without knowing in advance that they would be working on something that could one day be useful in terms of therapeutics. I think CRISPR is an example of that, a recent Nobel Prize discovery of mechanisms, bacterial mechanisms that underlie the ability to modify our genomes. I think the development of many drugs, so-called protease inhibitors, to help people who have HIV and AIDS is an example of that, based on years and years of people just being curious about how these enzymes work. That's really the underpinnings of all these therapeutics. So yes, and I think it was true for my graduate work, where I use yeast genetics to understand how hormones are made, and that some of that work ended up being useful for producing things like insulin in biofermenters. So you're absolutely right. I think that the most impactful discoveries in biomedical research often come from curiosity-driven research. Not all. Some are really directed at the get-go to important clinical goals, but many discoveries that sort of create a platform on which therapeutics are built really start from just curiosity-based research. And I'm so thankful that in this country we have institutions, public institutions like the National Institutes of Health and the UC system, that provide funding to me and other people like me who are just asking basic questions without knowing whether they will be quote unquote useful or translational at some point.
Thank you.
We have another question from Rajendra Pangini, and I apologize if I'm butchering that name. And the question is, how do you reflect your early career research life when you were a PhD and postdoctoral fellow? What key suggestions would you give to current early career scientists aspiring to spend their life in research?
Oh yeah, well, that's a very good question. I think going through graduate school and postdoc, it teaches you how to think about science, teaches you, of course, about certain techniques and how to carry out experiments. And you learn a lot. I was very fortunate to all through my career to have some amazing mentors who were really passionate about science and curious. I think the thing is that, you have to be patient with yourself in research. There's always, there are some very exciting peaks separated by long valleys where you're trying to figure out how to deal with problems. And a lot of what we do is not success, it's failure. And I think there's two things that have to keep you going. One is just a persistence. And with the idea that, if you keep working, you'll run into something and discover something interesting if you're asking interesting questions. And the other is that, you have to get enjoyment out of what you do every day, even if it's doing an experiment at the bench that you think was really a beautiful experiment, although it didn't necessarily answer the question you set out. You have to sort of take some enjoyment every day and sort of, I like doing experimental science because you get to work at the bench with your hands while you're also thinking. And that gives you an opportunity to really sort of enjoy what you're doing day to day, almost like a hobby. But in the long run, I think you have to really be interested in what you're doing and be driven by curiosity. And, we don't go into this, at least most of us don't go into this thinking we're going to win prizes. We just want to do this for the thrill of discovering something. There is a time when you make a discovery where you're the only person on the planet, or at least you think you're the only person on the planet, who knows the answer to a particular question. And that's a really thrilling moment. And I think that's what most of us live for.
That does sound like a thrilling moment to have the only answer to the question. We have another question from Laurel Oldock and her in the Q&A, and her question is, could you please speak to how heat and capsaicin, quite different stimuli, open the ion channel? What questions do the cryo-EM structures answer about this?
Oh, that's a very important question. So we understand more about how capsaicin, how a chemical agent activates this channel right now than how heat activates it. It's easier technically to understand how binding a chemical to a protein changes its shape. In this case, is an ion channel to open it up to allow ions to flow through it. And we have a pretty good sense of how that's done in some really wonderful work we've done with my colleague Yifan Cheng and members of our labs. There is some recent studies, not from my lab, but from other groups now who are trying to, and we're trying to do the same thing, trying to really look at structural changes that occur in these proteins with heat. But heat is a much more difficult stimulus to work with because it probably affects the protein in many different ways. And so we're working on that. But that's sort of the $64,000 question, as they say, in this field from a biophysical and structural point of view, how temperature can change the structure of one of these proteins to allow it to be activated, and how this relates to the ability of capsaicin or various inflammatory agents to do the same. So that's a great question, and I just say it's a work in progress.
All right, going back to the live questions, we have a question from Megan Multaney.
Yeah, hi, I'm with Stat News. Thanks for taking our questions. I just wanted to know, given that some of the other mechanisms for sensing the environment have already been recognized by the Nobel Committee back in the 60s and in the 2000s, for sight and smell and sound. What particular challenges did, sensing kind of heat and temperature and pressure present from a scientific standpoint that kind of, made it one of the last mechanisms in terms of our sensing environment to be?
Yeah, you're absolutely right. This has sort of been the last main sensory system to fall to molecular analysis. I think in part it's that we didn't really know what types of molecules to expect that we would find. There are differences, of course, in the visual system. You can do a lot of biochemistry because all of the mechanism is, at least for sensing light for photons, is located in one organ, the eyeball. So that's easier to study just from a technical perspective. And for things like smell, we've had some idea from pharmacology over the years what types of molecules might be involved. And so genetic methods to identify those by my former colleagues, Linda Buck and Richard Axel, there were more clues as to how to proceed. For things like temperature and pressure sensors, we really didn't have examples of the types of molecules that we could look for. And that's why Ardem and my lab had to turn to other ways to do this by functional screening or by using these natural products that we've used like capsaicin and menthol. So I think it was just not having a lot of prior information and some technical difficulties in terms of identifying and working with the nerve cells that carry out this process, which are sort of scattered over a much wider area in the body. And the other thing I should say is that working with stimuli such as pressure and temperature is in and of itself a little bit technically challenging in terms of carrying out these experiments. And so all of those things kind of made this a little bit less approachable than other sensory systems.
And Issam Ahmed had a second question, another question about timeline and applications. Do you want to go ahead and ask your question, Issam?
Oh, hi, can you hear me?
Yes.
Yeah, from what I understand, it was quite a process of trial and error to isolate the particular gene responsible for capsaicin and responsible for menthol. What made it so painstaking? Could you tell us a little bit about the timeline? And going back full circle, where do we stand now in terms of application in medicine? Thank you.
In terms of the painstaking aspect of it, I'd say that the most time was spent trying to figure out exactly what approach we would use to do this. Once we determined what approach was most likely to give us success, the screening for the gene was actually quite quick. And this was done by a wonderful former fellow in my lab named Michael Caterina, who's now on the faculty, has been on the faculty for years at Johns Hopkins School of Medicine. So once Mike and I settled on the right strategy, the screening actually only took a few weeks. So it sounds laborious the way it's described, but in actuality, due to some genetic trickery of molecular genetics, we were able to zero in on the gene encoding this receptor without too much flailing around, I should say. And then but as is true for all of this kind of research, everything seems to go fast at the end when you have the right strategy down. But there were at least 2 or 3 years of playing with other strategies that were failure at first. So it was sort of a little bit of a protracted time and then very quick to success. In terms of your second question, I think was about timeline to therapeutics. Is that correct?
He's muted again, but I believe that's correct.
Yes,
Okay.
yes.
You know, these receptors, many of them have now been identified and around for several years. In the case of the capsaicin receptor, over 20, and they have been subjected to a number of pharmaceutical firms for drug screening. And there have been some candidates that have come up. Some have tested out actually fairly well in some clinical trials for things like inflammatory pain. But in the case of the capsaicin receptor, there are actually some what I would call on-target side effects. And this sort of brings up one of the challenges of dealing in the pain field. And that is that aside from trying to achieve selectivity, that is hitting a target that's specific to the pain area so that you don't have side effects, you also have to walk this line of, excuse me, I just got to, okay. You also have to walk this line of wanting to inhibit pain that's chronic, so-called maladaptive, but not eliminate pain sensation that's protective or acute. So as we know, we need to be able to experience pain because that really keeps us healthy, because it prevents us from injuring ourselves or recognizing when we have or are about to injure ourselves. And we don't want to diminish that protective function. At the same time, we want to tap down these signaling pathways when they become hyperactivated in settings such as nerve injury or inflammation. And I think that's been some of the problem with early drugs, is that they turn off the receptor and they diminish people's ability to sense heat. So one worries about somebody burning themselves by drinking hot coffee because the drug is blocking their ability to sense noxious heat. There are people are now looking for drugs that have more subtle effects on the receptor and diminish their ability to be sensitized in the setting of inflammation, but not to block their baseline function as heat sensors. And I think the work that we're doing with EFON to really develop the atomic structure of these molecules and see what they look like in detail will really help to have a rational approach to developing those kinds of drugs.
We got another question in the chat. Could you, and this one's from Cynthia Gorney at National Geographic, and she's asking, could you elaborate on what made those experiments technically challenging? And just out of curiosity, where were you obtaining those menthols and chili heat sources? Are there lab versions of those substances?
Yeah, well, the nice thing about chilies and mints is you can just go buy them at the market. But most of these things, because we want to know exactly what's in them, we buy them from, a chemical company, from Sigma Chemical or something like that. But we did do experiments early on where we collected a bunch of peppers of different hotnesses according to Scoville scale, and showed that in fact, our assays reflected their hotness as you would detect that by putting them in your mouth. But why did this take a long time? Or why was this painstaking? Our goal was to identify a gene encoding something about which we knew nothing. So it wasn't like we had a protein sequence we could work from. We were sort of going blind, which means that we had an activity. We knew that capsaicin somehow activates this protein and should do something to a cell. And we had to figure out what that was and how we could set up a process, an assay, to detect that activity. So there were a lot of unknowns in doing this. We didn't know how many genes we would need to express in a cell to recapitulate this activity. So there were just a lot of unknowns. I think in the end we were lucky in the sense that some of the things worked in our favor. And once we understood how to do this, we were able to zero in on it. But the other thing too is that in order to do this kind of experiment, you have to sort through thousands of genes, which you can do by looking at even millions of genes, which you can do by sorting through thousands of genes at a time. But you have to have a process or an assay that allows you to look at genes in bulk and has the sensitivity to be able to identify what you're looking for. And that's part of what took us time, was to adjust our assays and the sensitivity of our methods so that we could detect very small responses in the beginning that would be diluted out by having many thousands of genes in our assays. So all of those things just took some time to work out. And as you often do in science, you work them out one little step at a time.
Okay, we've got another question from Rajendra Panjeni, and I really like this question. It is, so we learn pain, heat, cold stimuli, and our body response right from elementary schools. We grew up being fascinated about this. How do you explain your discovery to elementary school students?
That's a good question because I have to do that tomorrow. It turns out I have a session through the Gairdner Foundation with some 10 to 15-year-olds, and I have to tell them about what we've discovered. And, you know, I think for scientists working in certain areas, especially ones that are a little esoteric, that's always a tough job. For me, I think it's an easy job because I can talk about things that we all experience. You know, you put your hand on a hot stove or you put your hand on a block of ice. Now, getting to some of the molecular details is a little bit more difficult. But, you know, I show graphics and cartoons about what an ion channel looks like, what it does in a cell, how it activates a nerve fiber. And I think that's the most difficult part. But after that, you know, understanding what chili pepper is all about and what heat is about is something that we can all relate to. So I think I'm fortunate to work in an area that is sort of so approachable and immediately appreciated by everybody, you know, kids and adults, et cetera, because it tells us about the world we live in. I think that's the great thing about sensory systems. You know, sensory systems report to our brain what our world looks like. And we appreciate the world through all our sensory systems, the colors we see, the sounds we hear, the textures we touch, or the temperatures that we touch. So we can all relate to that. It's a part of biology that's so relatable to everybody.
Okay, well, we've got time for one more question, and it looks like Tina Say she has her hand up again. Tina, did you have a follow-up question?
I do. I was wondering if there are people who have mutations in either of these receptors that you discovered that would change their perception of heat and cold.
That's a very good question. Mutations in these genes are apparently quite rare, or they're not so obvious that people come into the clinic with deficits. There is a report that there are mutations in, or I should say variants in gene encoding the menthol receptor, not in the region that encodes the protein, but in regions that control its expression, and that there are differences depending upon where people live, say in northern cold climates or southern warm climates. And it's presumed that this accounts for their differential ability to interact with their environment in terms of ambient temperature. There are some recent identification, I believe, of people who have mutations in the capsaicin receptor, and I'll be interested to see what they look like when they're examined by neurologists. There are mutations in, you know, in Ardem's work on touch sensation. There are some mutations in those channels that have very interesting effects that are quite profound, and so they're identifiable in the clinic. So there are mutations in different proteins in the touch and pain pathway that diminish people's sensitivity to some in pain, and they've told us a lot about how the pain pathway works. But in terms of the proteins that we've looked at, there aren't a lot of mutations that have so far been identified. But those would, of course, be fascinating to look at. And much of what we surmised about their role in mammals comes from looking at mutations in, for example, laboratory mice.
Okay. Let's see. Okay, there's two more questions, and then we'll have to end this fascinating conversation. But the second to last question is from Mohammed Mansour, and his question is: he's a science journalist from Cairo, working for Al Sharq News. And he says, I would like to ask, what are the implications of the discovery that have been for understanding the molecular basics for this sensation of heat and pain?
So the implications from this are, one, just understanding it. I mean, that's our main goal, is to understand how physiology and how the body works. But the other implication is that each of these pathways, our ability to sense heat, our ability to sense cold, becomes dysregulated when there's injury. So for example, when somebody's treated, many people are treated with certain chemotherapeutic agents when they have cancer, they become moderately sensitive to cool temperatures, and this and anything that's mildly cool then gets sensed as extremely painfully cold. And so there are a number of people who are looking at the cold receptor and the cells that express these receptors, because they are the genes that we've cloned not only identify the proteins, but they identify the cells that are important in temperature sensation. And this identifies molecular pathways that could possibly use to treat this situation, which we call cold allodynia. And the same thing's true in the area of the capsaicin receptor in heat, where dysregulation of that pathway leads to hypersensitivity to temperature in conditions where there's inflammation, such as when you get a sunburn or when you have arthritic pain. And so understanding the molecules involved and the cell types involved gives us clues as to how we can target these pathways to develop drugs that can be used for very specific types of pain situations that lead to hypersensitivity to temperature, or in some cases to touch.
There was one last question from Laura Oldock, and that was, what is the most beautiful experiment you've ever done? It seems like a great ending question for this.
The most beautiful experiment. I always say the most beautiful experiments I've ever done are the ones I did when I was a graduate student, because I did them with my own hands. But and here I have to say that I have not done these experiments that we're talking about in relation to this prize. It's been done by some unbelievable people who I've worked with, students and fellows. I can't single out any particular one because I don't want to favor anyone. But of course, cloning of these genes of the capsaicin receptor by Mike Caterina and of the cold receptor by David McKemie and Werner Neuhauser when they were in my lab are really sort of foundational experiments and thrilling moments. Other thrilling moments, however, are, for example, work we've done with with Yifan Cheng and Erhu Cao in my lab, who was a postdoc, and Maofu Liao in Yifan's lab, when we saw the first atomic structures, as Sam alluded to, by this method called electron cryomicroscopy. That was really a beautiful moment to see the molecule that you've been thinking about and working on for some 20 years and actually seeing it in the flesh, as they say. And that was also a thrilling moment for this technique of cryo-EM, which really represented sort of a landmark moment in that field, being able to take resolution of proteins to the atomic scale. So I would say those three things are thrilling, but so many people in my lab have done beautiful experiments that, I'm so proud about and that have helped to push the field forward. So it's hard to pick a favorite, but those three stand as, I think, examples that come to mind.
Okay, well, thanks so much for your time.
Thank you.
Morning, David. And we're gonna wrap things up and give you a little bit of a break. We've had a long morning so far.
A long morning so far. Yeah.
Thanks everyone for attending, and to Sam and everybody for being here, Sam and Talmadge. And we are recording this, and it will be available online for everybody at ucsf.edu. So feel free to revisit it.
Thank you, everyone. I look forward to seeing my colleagues on campus today.
And congratulations, David.
Thank you so much. Bye-bye.
All right.
Thank you, Robin.
All.